Today, I will discuss part 2 of cell division. The topics include cytokinesis, cleavage furrow, and cell plate, contact inhibition, meiosis, synapses and chiasmata, crossing over, and the comparison of metaphase of mitosis and meiosis. To start off, we have cytokinesis, which is the dividing of the cytoplasm. It is different in plant and animal cells. In an animal cell, it is called a cleavage furrow. In a plant cell, it is called a cell plate. Next up, we have a cleavage furrow and cell plate. Cleavage furrow forms down the middle of dividing animal cells only. Actin and myosin microfilaments pinch in the cytoplasm, enabling the cell to break in two. Cell plate forms down the middle of dividing plant cells only. It is formed by a fusion of vesicles from the Golgi apparatus. Next up, we have contact inhibition, which is a phenomenon in which cells cease dividing when they become crowded. It is also called density-dependent inhibition. It is a property of normal cells, and cancer cells do not exhibit this characteristic as their growth is uncontrolled. Next, we have meiosis, which produces gametes and gonads. It consists of two divisions, meiosis I and meiosis II. Meiosis I, which is reduction division, is where synapses and crossover occur. Homologous chromosomes also separate during meiosis I. Meiosis II is like mitosis in that chromosome number remains the same. Sister chromatids separate, they uncouple at the centromere. Next, we have synapses and chiasmata. Synapsis occurs during the prophase of meiosis I. Homologous chromosomes pair up in a tetrad bivalent complex. It is the preparation for crossover. Chiasmata is the site of crossover for non-sister chromatids. It appears as an X-shaped region under the microscope. Next, we have crossing over, which occurs during the prophase I of meiosis. It is an exchange of genetic material between non-sister chromatids. It increases variation in offspring. It produces recombinant chromosomes consisting of combined genes from two parents. Crossing over occurs after synapsis. And lastly, we have the comparison of metaphase of mitosis and meiosis. In mitosis, sister chromatids separate. In meiosis I, homologous pairs separate. Mitotic daughter cells are identical to each other and to the parent cell. Because of the exchange of genetic material during meiosis, or crossing over, gametes are very different from each other.